We have a few minutes for questions. Are there questions? Okay, Rosen.
Thank you for a great session. So my question is really to Lucy, both you, the whole team. I think one of the biggest questions when you think about formulas, when you think about failure of formulas, and you quote the Europreval studies, is that it was driven, the failure was by non-IgE mediated allergy. So, and Claire, the data that you've presented is really epitope data based on IgE mediated allergy. And it doesn't matter which publication you look at, the failure that is described is not an IgE-mediated allergy, it's a non-IgE mediated allergy. So my question is, whilst you in vitro show differences, the reality is clinically in a non-IgE mediated patient, I can tell you I see no difference between the different hydrolysates.
I believe this is a part of clinician part. I believe that we have to decide which non-IgE mediated clinical manifestation we are speaking about. As in my experience, in severe non-IgE mediated cow's milk allergy, the children recognize the let's say, less extensive hydrolyzed formulas. So this is a question that we have tackled for, I said before, 20 years. Now we have something sound, and I would advocate and would like to ask Claire if we can have, let's say, a repository for the formulas, extensive hydrolyzed formulas, as these are actually the only therapy for these children. So if you have a drug in a pharmaceutical world, you have a list of the adverse reaction on how the composition is. So I would advocate for treating these formulas the same way we have for pharmaceutical drugs, as they are drugs, treatment, the only treatment. So I ask Claire if she can comment on that.
So, is this on? Yes, this is now. So, it's a really good point. And of course, if you've got a cell-mediated reaction, which is at the basis of your non-IgE, we actually haven't defined what the triggers are. Are we talking about a 9-amino acid T-cell epitope, or are we talking about something different? How do they overlap? In fact, to be frank, we have very few studies that even show what the T-cell epitopes are in IgE-mediated allergy. There are handfuls of publications on this, and ensuring that they're the same in different populations around the world, or do they change and especially with infants, because the picture changes so quickly as those young children, infants, grow up. So I think actually we really should identify what the triggers actually are in the non-IgE cow's milk allergy. That would be really helpful. It would then also help us to say, well, are we targeting the right peptides? And we have lots of reagents that can be used to manufacture hydrolysates, because if you can get the target, I think they can get rid of it. So that would be one thing. And it may also be that the EHF8 that has hardly any in, maybe that would be suitable for the non-IgE kids, or 7 and 8, and maybe the ones in between aren't. But that's where the clinical study really becomes very important. But I can see that, and discussions I had last night is like the non-IgE is really important. So I don't know if that answers Antonella's question, but I hope it addresses what Roseanne raised.
I mean, I was going to say, from my perspective as a dietitian, obviously like Roseanne as well, I think I have seen differences in patients with the extensively hydrolyzed formulas. I'd say it would be more based on the types of proteins, whether it was casein or whey. And again, it's hard to know whether that's because one is maybe a bit more broken down than the other, or whether it's that particular protein that is the problem for that infant. So I think, the non-IgE mediated allergies are sometimes more difficult to kind of manage and kind of get on top of, because of this sort of very non-specific symptoms that are just infant infant symptoms as well. So I think that's where it's really difficult to be sure. And also you've got the parents that are extremely anxious and, want to see those changes very quickly. So, maybe a bit longer would have resolved the problem before moving on to a different formula. Yeah.
Other questions? Other questions? So I have a question for you. It is difficult to manage these children, as sometimes clinicians prescribe the formula and then they don't see them anymore. When and how do you believe it would be important to evaluate the nutritional requirements and if the guy, the boy, the girl are in an elimination diet, you have how you can check if he meets the nutritional requirements?
Yeah, so I mean, if an infant is, fully formula fed, the first 6 months of life, this will be their only source of nutrition. And so we know the infant requirements, around sort of 150 mils per kilo in that patient group. And so if they're meeting that volume, they'll be meeting their energy, their protein, their fat, their micronutrient profile. Obviously, over that time, you'd be monitoring their weight and their growth, head circumference as well. So we're not just looking at individual weight measurements or individual length measurements or head circumference. They need to be plotted and looking at that trajectory. Because if you're not looking at the trajectory, an individual point might seem fine on the growth chart, but actually if you were to plot everything else, hold on, they've suddenly dropped down 2 centiles. So we need to be monitoring their growth and development. In terms of when they start on solids, the majority of children with a cow's milk allergy will wean onto a milk-free diet. And again, we continue infant formula alongside solids until 12 months of age, but as the oral diet progresses, we'll start to reduce the amount of formula. And so it's so important that they're using sort of fortified plant-based alternatives, that you're sort of counselling and advising the families on sort of key micronutrients such as B12, calcium, vitamin D, iodine, if we're avoiding, cow's milk protein in particular, and actually particularly if they have other exclusions such as fish and egg as well. Iodine's the one we're really going to be looking at in those patients. I wouldn't do any blood markers personally in my clinical practice unless there was a clinical reason for that. So signs of malabsorption, unexplained weight loss or poor weight gain that, they seem to be meeting what they need, but they're just not meeting those targets.
So would you consider feasible that any time a pediatrician diagnoses cow's milk allergy and prescribe a formula, an extensive hydrolyzed formula, together with an elimination diet, would it be feasible to schedule a regular visit with the specific devoted dietitian, or according to the context, what could be the minimum time for reevaluating?
Yeah, dietitian. Yeah, so I mean, if you've got, like I say, if you've got a patient who's growing well, they're following the diet, the parents are understanding the information, you might see them maybe at sort of 6 months to support with weaning, and then at sort of 10 to 12 months to support with reintroduction. It really does depend on a case-by-case, reason as to why you might see them a bit more frequently, but I'd say probably 3 to 6 months would be appropriate in most cases.
Thank you. And Claire, can I ask you if you consider that we should have guidelines for analytical process of extensive hydrolyzed formula that should be, let's say, submitted to EFSA as, let's say, regular mandatory requirements for putting in the market an extensive hydrolyzed formula?
So I think it would really help everybody if we had a kind of like, if you like, a specification of not just what the size distribution is, but the proportions of what is left, and taking account of aggregates. And I think the technology that we now have means that you could have the analytical methodology available to back that up. And it would, I think actually it would get rid of, I mean, I was shocked at the variation that we saw, to be perfectly honest, Antonella. And I think some of those they just wouldn't be allowed on the market. It would improve the quality generally.
Thank you. So, other questions? Josephine?
Hi. It's not really a question, it's more a comment. I think we have guidelines because we have the hypo. If you follow the AAP guidelines and you prove that your product is hypoallergenic, I mean, it's blinded, it should be okay. And I think that's the criteria that all companies should follow.
Yes.
Can I just say something there? It depends how you do your analysis. And if you do your analysis in one way, I can tell you they all look the same. And it's because of the analytical tools and what they're actually looking for. And you do need some kind of methodological approach that backs that up. Because I think the current ones are too woolly from that perspective.
But the thing about the hypo studies is it's tested in actual patients in a blinded manner. So,
but they can't use that to monitor every single batch. You need test methodology that you can deploy in your quality control program, and that is what this methodology would do.
yeah. Okay, so in addition to the hypo in patients, then you say that each batch needs to be tested. I thought batches were tested by
Yes,
companies.
but I can tell you.
That's the point I'm trying to make.
Yeah.
It depends on the methodology that you use, okay?
Okay,
And
clear,
maybe
yeah.
there are inadequacies in those methodologies, and it is not, the methodology is not specified by the regulatory authorities.
Thank you.
For sure, testing in vivo is paramount, but I can tell you that, as Rosan said before, some children can tolerate, some others not. So when we started the journey with Susanne Alken and the whole group, I would say 20 years ago, which is really shameful, the issue was that looking at the labeling in each extensive hydrolyzed formula, some had ultrafiltration, some mentioned enzymatic analysis, some others whatever. So they were different from each other, first. Secondly, some brands put on the market used as references studies performed by other brands. So the point is, can we put order as a nice housewives in this mess? I think that it is timing for doing that, as we cannot leave such uncertainty in our patients, in our children, considering also that some of these formulas are reimbursed by the national health system. For what? Sometimes they are not serving the task they are prepared for. So I believe that this would matter for further discussion, hopefully not for the next 20 years, and we would like to come to the goal where we give the right formula to the right children at the right timing. So I would like to thank you very much for attending this session, inviting you to the next session kindly sponsored by Nestlé for an update on what Claire said to us today. Thank you.